Zone, and today we're going to do another set of notes, and this particular set of notes is on cell division. Please have your Cornell note paper in front of you and write in the topic objective cell division. We have two separate essential questions today for the purposes of these notes, and the first one is what events take place during the 3 stages of the cell cycle. And the second essential question is how does the structure of DNA help account for the way in which DNA copies itself? So the way DNA is set up makes it so it's a lot easier for it to replicate or copy itself. First, we're going to start with the 3 stages of cell division. So in the notes section, you want to make a heading of the 3 stages of cell division: interphase, mitosis, and cytokinesis. The first we're going to start with is something called interphase, which is the first stage of cytokinesis, the first stage of cell division. First, we need to identify what the cell cycle is. The cell cycle is just the regular sequence of growth and division that cells undergo. We've talked about before, when you get bigger as you're growing, if you get a cut, anything that happens in your life that it's going to require cells to grow is going to include the cell cycle. This is just the normal process that cells undergo. And that first stage is interphase, and the cell actually spends most of its time in interphase. During interphase, this is the period actually before the cell divides, and during this phase, the cell grows, makes a copy of its DNA, and prepares to divide into 2 cells. So interphase is the period before cell division where the cell grows, makes a copy of its DNA, and prepares to divide into 2 cells. The first part is growing. When the cell is growing, the cell grows to its full size and produces structures it needs, organelles such as ribosomes, for example. So here I have a little picture of a cell right here. This is clearly an animal cell. You can see the nucleus, you can see some mitochondria over here, and other organelles within in the cell. Next, the cell needs to make a copy of its DNA. If it's going to split into 2, it needs to make sure it has the same instructions in each half that it splits into. So it has to copy its DNA. The cell makes an exact copy of the DNA in its nucleus in a process that is called replication. At the end of these notes, we're going to get into replication a little bit more, but I'll just put a little picture here. Here you have your DNA, kind of looks like a twisted ladder, and it unwinds and unzips and makes a new copy of itself. And again, I'm going to get into that more towards the end of the notes. Okay. Next, the cell prepares to divide. So it's getting ready to split into 2. And how does it do this? Well, it produces structures. Oops, sorry. It does this by producing structures it will use to divide into 2 new cells. And I'm not going to really get into that. But it makes little things on the sides of the cell that help it pull it apart into 2, basically. So here's a little picture. It's preparing for division. probably not the same kind of division that this guy's getting ready to do, although, assuming he was a real person and not a cartoon, his cells would be going through this as well. Okay, so we've done stage 1, interphase, which is the part of the cell cycle that the cell spends most of its time in. It's preparing for division by growing, making new structures, and copying its DNA. And now we're ready to start dividing. And that is called stage 2, and it is mitosis. Let's define mitosis. Mitosis is a stage during which the cell's nucleus divides into 2 new nuclei. One copy of the DNA is going to be distributed into each of the 2 new daughter cells. So remember, one cell is making a copy of its stuff, and then it's going to split it up in between 2 new cells. And that's how, humans grow. Okay, here we go. Here's your definition. The stage during which the cell's nucleus divides into 2 new nuclei. There are 4 parts of mitosis: prophase, metaphase, anaphase, and telophase, otherwise known as PMAT. It's a good little mnemonic to remember it. Prophase, metaphase, anaphase, telophase, PMAT. Let's start with prophase. Okay, prophase is the first stage of mitosis. And during this part of mitosis, the chromatin, remember in the nucleus of the cell, the DNA is these little thread-like structures known as chromatin or chromatin. But during prophase, the chromatin condenses into these nice little double rod structures called chromosomes. You've probably heard of chromosomes. Each rod is an exact copy of the other, so I'll show you a little picture of that. So here we go. We have a cell. And here's the chromosomes. Each little side of it is known as a chromatid, and together they are sister chromatids. So this is one sister chromatid, this is the other, and they're held together by something called a centromere in the middle. I'll give you that little information under here too. I apologize, I'm not well. Okay, chromatids are the 2 rods that make up a chromosome. So again, here's a chromosome, and there's 2 separate rods. Each rod contains an exact copy of the other rod's DNA, and they're held together by something called a centromere. Okay, so that's prophase. You have the chromatin condensing and forming chromosomes that are an exact copy. Each side of the chromosome is an exact copy of the other. Now let's move on to the next phase. So, and that's called metaphase. In metaphase, the chromosomes now line up across the center of the cell, and each chromosome attaches to a spindle fiber on either side of the centromere. I'll show you what that is, because that sounds confusing. So here you have the chromosomes have now all lined up in the center of the cell. And so you can see that they're attached in the middle, and then they're attached on the side by these little fibers that are going to end up pulling them apart. And that's how you're going to end up with this side getting the same kind of DNA and the same information as this side. They're both going to get exact copies. Next, we have anaphase, and that's where that pulling apart actually happens. So in anaphase, the centromeres, the little thing holding the two parts of the chromosome together, those centromeres split. And so then the two chromatids, remember, each side of the chromosome is called a chromatid. So when the centromeres split, the chromatids are going to split too. And then they're going to be pulled apart and pulled to the opposite sides of the cell by these fibers here, called spindle fibers. So here we have a little picture of that happening. So they've split, and now they're being pulled to either side of the cell. Next, we have telophase. Telophase is the last part of mitosis, and then we're going to move on to something else. Telophase is the chromosomes begin to stretch out. Remember, they're in these rod-like structures right now, but they want to go back to their state of being like chromatin, like the uncondensed version. So the chromosomes begin to stretch out, they lose their rod-like appearance, and then a nuclear envelope forms around each region of the chromosomes. So remember, you have two so now remember, these were pulled apart to either side of the cell, and now the new nuclear envelope is forming around the chromosomes or the chromatin on either side of the cell. Okay, and that's mitosis. If you have ever seen the cinematic or cinematic masterpiece that is Twilight, you might remember when Edward and Bella are in science class and they're looking through the microscope at the stages of mitosis, and they're saying anaphase, prophase, metaphase. And now you know what that is. You can go back and rewatch that movie. Actually, don't rewatch that movie. It's terrible. Okay. Stage 3. So again, we have interphase. The cell has, stage 1 was interphase. The cell is growing and making all the structures it needs. It's making a copy of its DNA. Then mitosis is happening. The copies of the DNA meet up and they get into pairs and they line up across the cell and they get pulled to opposite sides, and now you have two sections of new nuclei really in the cell, but now the cell needs to split into two, and that's stage 3, and that's something called cytokinesis. So this is the final the final stage of the cell, where the cytoplasm divides into two and the organelles are distributed to each of the two new cells. So you end up with two new cells. They're known as daughter cells, and they have formed from the original cell at the very beginning. And they're identical to the original cell as far as their DNA makeup is concerned. It is possible that one side of the cell, one daughter cell gets more of one type of organelle than the other. It doesn't always happen perfectly evenly, but they have the exact same DNA, so they're going to have the same information to carry out whatever the cell needs to do. So I have a little picture of that. So here we have two new cells. They're the daughter cells, and their chromosomes have unwound themselves back into the chromatin, and they have two identical nuclei with two identical sets of information. And now, what are each of these daughter cells going to do? Well, they're going to go right into interphase, and they're going to begin again with a new cycle. The cycle will continue with each of the daughter cells. So yeah, now each of the daughter cells will go into interphase and the cycle will continue. Okay, now we're going to talk about something really exciting and really confusing. And I apologize that I'm not there. I promise you we're going to do lots of stuff in class that that's going to hopefully help you understand how DNA is structured and how it ends up replicating itself. And we're going to do a little activity, hopefully on Wednesday, that goes through the cell cycle. Okay, let's talk about the structure and replication of DNA. So DNA looks like a twisted ladder. Here we go. I'm going to give you a little picture of that. Here we have the twisted ladder. The sides right here, so here's our twisted ladder. The sides are made up with sugar molecules that alternate with phosphate molecules. The sugar is called deoxyribose. DNA stands for deoxyribonucleic acid. Okay, so you have the sides of the ladder. The sides are made up of the sugar and phosphate molecules, and then each rung of the ladder, so each of these little rungs is made up of a pair of molecules. You can see they're two different colors on either side. And these are called nitrogen bases, remember? So deoxyribonucleic. So this is, we already talked about the deoxyribo, which is the sugar. Nucleic has to do with these nitrogen bases. DNA has 4 kinds of nitrogen bases: adenine, thymine, guanine, and cytosine, and they're represented by the letters A, T, C, and G. And the way it works is kind of nifty, and it makes it really easy for replication. And that is that A always wants to be with T, and C always wants to be with G. So if you look up here, you see the green and the blue always line up no matter where they are. And the purple, which is in the orange, always line up. They always want to be together. It just happens that way. You can make up a little mnemonic to understand this. I don't know. So I don't remember what other people have come up with, but maybe we can do that in class together on Wednesday. How to remember A, T, C, and G. But how does it work? So what happens during the replication process? Well, first, the 2 sides of the DNA molecule need to unwind and then separate, like a zipper unzipping. And I know we had this picture at the beginning, so let's show it again. So here you have the 2 sides. They've unwound and they've unzipped. And you can see something happening in there, and I'm going to explain that a little further. So when they unzip the 2 nitrogen bases, they separate, okay? So if you had an A and a T here, they've now separated. And in the nucleus of the cell, there are other nitrogen bases kind of just floating around. And so new bases pair up with the bases on each old strand, and they form new strands. And the new strands happen to be identical to the other ones. So let me see if I can make that. Oh. Okay. So if you had an A over here and a T over there, and you had these free nucleotides floating around, so if you had an A here, then a T is going to find it, and if you had a T here, then an A is going to find it. So when you're done, you end up with 2 identical strands of DNA, and that is how DNA replication occurs. I promise you we're going to get into this a little bit further in class. We're going to practice it. We're going to do some analogies and make little metaphors, and hopefully you'll really understand it by the time we get done. But for now, I'm just going to stop there. Please don't forget to write a conclusion. Make sure you answer the two the two essential questions. Thanks, guys.